All right, so we're going to start off with notes on photosynthesis for this week. Since you already did your lab, you kind of already have your basics down, and you should know some stuff from your biology class that you've taken before. If it's been a while, that's okay. We're going to go over it in more detail, but you already know at least the equation for photosynthesis. And so for your do now, I'm going to ask that you pause right after I explain it and actually try to do what it says. So describe what you see happening in photosystem 2. This is this diagram shown right here on the right. This is the first part of the process of photosynthesis. So it's a lot more detailed than you might have known already, but just kind of describe in 1 or 2 sentences what is happening in this diagram. All right, so after you've done that, the agenda today, obviously, like I said, is photosynthesis notes, and then your chapter 8 summary and Learn Smart are already posted, which means that you need to get them done. Since Thanksgiving break does start after Friday class, I'm not going to be assigning you this overlapping with something else. You do have a break assignment, but this assignment for the chapter 8 summary and Learn Smart needs to be due by Friday, end of day. Okay, just to keep that in mind as you're working. Your learning objective is the same as we've done before, but the focus today is on the capturing and storing of free energy. So when we talk about use, that will be on cellular respiration. So this is the same diagram that you were just looking at, and it's broken down kind of into 2 parts. This one is for right here, and then all of this right here is about here. Before I start talking about anything, I do want to let you know that the vocabulary I'm going to be using is chemistry related for these 2 terms. So when something or a molecule or a chemical gets reduced, it means that it got an electron added to it. And when it gets oxidized, it means that an electron was taken from it. Okay, taken and put somewhere else, taken and used for something else. Okay, so write these down on the side just so that you have those notes as reference in case when I say the words, you're like, what the heck does that mean? And then you can look at it and then refer to it. If you've already taken chemistry recently and you know it, that's even better. You won't even have to look at those. So in photosystem 2, it's kind of weird because I know number 2 is usually like, step 2, second part, but in photosystem 2 is actually the first system that reacts and does anything during photosynthesis. And this is just because it's only called photosystem 2 because it was discovered second. So photosystem 1 was discovered first, but it actually is the second step of the process, while photosystem 2 is something that got discovered later on, but is actually the first step of the process. So in photosystem 2, this is kind of probably a little bit more complex than what you already said, but it's the same prompt as what you just saw in the do now. So the sun provides the energy to extract electrons from water. So you can see water down here, and there's an electron that gets taken out or extracted. Okay, and that's the sun energy doing that. And that splits the water into 2 hydrogen ions and half of an oxygen molecule. So only when you do even numbers of water at a time will you actually get full oxygen molecule. So this is kind of important right now that you know that it's only half of an oxygen molecule for each electron that's produced, right? Because there's only 1 O. And the hydrogen ions aren't bonding to anything. They're just there. And remember, when there's a lot of hydrogen ions, something is acidic, right? Because it's related to pH. So the electron gets energized in the chlorophyll pigment complex. And you don't need to know details about the pigment complex, except for the fact that it's part of the chloroplast, and on the inside, it's part of the grana and thylakoids, okay, which are these stacks. So the energized electrons then travel in the electron transport chain, which brings it over to photosystem 1. And in the diagram here, it might be a little confusing, but the image isn't showing you that the electrons are going closer to the sun physically. It's along this arrow right here, which is showing that the electron is accepting energy and it's increasing in energy level, and that's it. Okay, it's not saying that it's going anywhere. It's still here inside of the system, inside of the chloroplast, inside of the plant. Okay, so overall, again, photosystem 2 is capturing light energy from the sun. It causes an electron to be ejected from the reaction center. So the electron is actually going to leave here after it gains enough energy, and then it'll go move down the electron transport chain to photosystem 1. Okay, the electron that is leaving here is going to be replaced with another electron from water, right? So another water gets split and it forms 2 more H+ ions and another O. So once you get that second one, this oxygen, those 2 oxygens will combine together to make 1 full oxygen molecule, which is O2. Okay. And then as it's going through the electron transport chain and moving and losing energy doing reactions, it's actually causing the hydrogen ions to accumulate within the thylakoid. So within one of these little pancakes, the inside is going to get filled with a lot and a lot and a lot of hydrogen positively charged ions. And then this ion gradient of hydrogen ions is going to eventually be used to produce ATP. There's an enzyme that does it, but you have to have a lot of H pluses on one side of a membrane in order to force the production of ATP. Oh yeah, and also right here, if you notice, hydrogen Plus is also interchangeably used with the word proton or hydrogen ion. Okay, so because all of them have the same mass and the same purpose, right? They interact the same with all other molecules because it's just a positive charge with almost no significant mass. Okay, so in the light reaction in photosystem 1, the electrons from the electron transport chain get re-energized by solar energy. So again, light energy is hitting all parts of the plant and all parts of its cell at all times. So it's getting re-energized again in another pigment complex. Okay, and this is again in chlorophyll. And those electrons, along with the hydrogen ions, are going to reduce or add an electron to NADP+. And when you add this electron, it went from here, from the water, it got energized and moved through the electron transport chain to make ATP. You're going to have it over here and it has to get re-energized because right now it's low energy again because all of its energy got used up to produce ATP right here. So it gets re-energized still by the same sunlight that's constantly on the plant. And that electron is going to get added to NADP+. Remember, that's why NADP+ is getting reduced because an electron is getting added to it. Okay, so you're reducing NADP+. So when you add the electron and then you also add the hydrogen that's just floating around, the hydrogen ions, the protons, you're going to end up with a molecule called NADPH. And this is literally just an electron shuttle or an electron carrier. So these electrons that are now added to it are high energy electrons and they're going to be used later. But you can't just have electrons free flowing everywhere because they have a charge, they'll react with a bunch of other things. So when you put it onto the NADP+, along with the hydrogen ion, you get a secure molecule for now where it's stable and you can move it around to where it needs to go. And then you'll react it with something that likes the electrons even more. So it'll tear the electrons away. So this NADPH is an electron carrier. It literally acts like a shuttle bus, right? It's going to take this electron from here to its second destination as a shuttle bus, like any kind of bus does. So overall, the electron transport chain, and we'll refer to the electron transport chain as the ETC a lot of the times just because it's easier to write or easier to say. So the ETC is in charge of producing the hydrogen plus gradient. Remember, it means that one side of the membrane is going to have a lot more hydrogen protons than the other side of the membrane. And as these hydrogen protons flow back down the concentration gradient, so they're going from the high to the low, they're going to do it through an enzyme called ATP synthase right here. So as you can see on the inside of the thylakoid, there are a lot of protons. And it's not really a sustainable thing because the membrane is semi-permeable, right? So they're going to want to even out the gradient into an even distribution of protons on the inside and the outside of the thylakoid. And the easy way for it to do that is to go through this protein channel. The thing about this protein channel is that it's also an ATP synthase, and synthase or synthesizing, it means that it's going to be making ATP. And remember, it's an ASE, which means that it's an enzyme, right? It's a protein that has a function in metabolism. So here it's going to be, as the protons are moving through it, it's going to be taking a P and an ADP, an adenosine diphosphate, and it's going to use the energy of the movement of the protein, of the proton, to then create an ATP out of these reactants. Okay, so this is called chemiosmosis because molecules are moving in osmosis and it's changing or affecting chemicals or building or breaking chemicals, and here it's building. Okay, so overall in photosystem 2, electron transport chain is producing ATP through chemiosmosis. So this whole thing right here that we've talked about is the first half of photosynthesis. We'll talk about the Calvin cycle right after this. But the first half of photosynthesis is when the sun provides the energy to break water into hydrogen ions that remain in the thylakoid space, and oxygen gas then diffuses out. So oxygen gas is produced during this first part of photosynthesis. We haven't produced anything else yet. We haven't produced the glucose that we're looking for, just the oxygen gas because it's coming from the water. Okay, the energized electrons then pass from protein to protein in the electron transport chain, providing the energy to pump the hydrogen ions into the thylakoid space. So it's going to fill this space with hydrogen ions, and it creates that concentration gradient that you need for ATP synthase. So as the hydrogen ions then flow down their concentration gradient through ATP synthase, that provides the energy to make the ATP. Okay. Also, the electrons from photosystem 2 get re-energized by solar energy in photosystem 1, but this time the electrons end up reducing NADP+ into NADPH in order to carry those electrons over to the electron transport chain. So the next part, like I said, is going to be the light independent reaction. So when we talked about photosystem 1 and 2 in the first part of photosynthesis, they both use light energy from the sun. that being said, the second part of photosynthesis doesn't actually require any sunlight or any sun energy anymore because the electrons that are going to be used are already energized, right? And then they're being carried over. So for the Calvin cycle, what is being consumed is carbon dioxide here. So we know that water got consumed in the first part and oxygen was produced. Here we are consuming the carbon dioxide from the atmosphere now. And what is being produced is going to be glucose and phosphate, okay? So, sorry, not glucose and phosphate. It's going to be a molecule called glucose phosphate, right? It's just one combined molecule. And we're going to refer to it as G3P because there's going to be 3 carbons in it and it's going to have a phosphate attached to it. So what is going to be regenerated? This enzyme called RUBP, okay? It's being used in the cycle, but then it's going to be unaffected by the end of it so that it can be continually reused and reused and reused in every single cycle as long as it's free. Okay, this is what RUBP really is, but we're not going to get into the details of that. You just need to know that it's reused and in that it's not ever broken down or changed. It's an enzyme, right? It's going to be reused multiple times every time there is a cycle in the Calvin cycle. So as you can see, carbon is going in, and we're going to have 3 carbon dioxide molecules go in at a time because we need to make at least a molecule that has 3 carbons in it. And then when you combine 2 of those, you get glucose because that's a 6 carbon molecule. So there are going to be 3 carbons used during carbon fixation. So you're going to change it from gas into a chemical form that can be physically solid. And then so there's going to be multiple of these molecules that are created during carbon fixation, and they're going to get reduced. And during reduction, the ATP right here, the energy is going to be used up, okay? The Calvin cycle is an energy using process, although overall, there's a balance on how much ATP is used and how much ATP is actually created. Because remember, we said earlier in the ETC, in the electron transport chain, that ATP is actually being made as well during photosynthesis. It's just getting balanced out because it's also being used. Okay, so you're going to use up a certain amount of ATP, and you're going to be getting another intermediate carbon molecule that's not our final product yet. And then you'll use the electrons from NADPH. Right, we talked about that earlier. It's an electron carrier. It's going to get actually used up now. So the electrons are going to be added to the intermediate product, and then you're going to actually get G3P. And G3P is what we were aiming for as our product towards the end of the Calvin cycle, because then you can combine G3Ps to make glucose. Okay, you can also make it into other organic molecules that require carbon as well, which is why it's a super useful molecule. It's a basic building block molecule for carbohydrates. Okay. So afterwards, parts of the leftover are going to be then affected by ATP again, and then the energy is going to help return the enzyme RUBP into a usable form. So in the beginning, right here, when we're fixing the carbon dioxide, you're going to be using RUBP, and all these intermediates technically still have RUBP, the molecule, built into them and hidden inside of them. But when you want your end product, you want G3P, you just have 3 carbons and a phosphate. You're not going to want the RUBP to leave with the cycle because you're not using it for anything else in photosynthesis to build any molecules. So you have to return it. And you return it by adding the energy to break and make some more bonds, and you're going to be returning RUBP to its original form. Okay, and then that RUBP will go again to continue fixating some more carbon dioxide from the atmosphere. So what is getting oxidized here is the NADPH. The electrons are being taken away from the NADPH right here and added to the carbon molecule intermediate to make G3P, right? The electrons originally, NADP plus was originally reduced in photosystem 1, but now it's being oxidized again. So when you have the NADP plus here and it's oxidized, it's going to go back to photosystem 1 and do its job again. It's going to carry back and forth, back and forth, back and forth. What is being reduced? The carbon dioxide right here. It gets reduced originally and it gets electrons added to it during its combining with RUBP. And as it goes through, it's going to be, it's getting the electrons added to it right here. So the electrons are getting added to it. So the NADPH is losing the electrons, and then the G3P, or what used to be carbon dioxide, is going to be gaining that electron to make that final product. Okay, so the importance of the Calvin cycle is that G3P can be converted into a lot of things, like I said just now. It's going to be either converted into glucose or other organic molecules that require carbon. So we have fatty acids, we have glycerol, which is part of so we know that biomolecules, that fat lipids are shaped in the shape of an E, a capital E, and that combiner on the side that has the joints for the 1, 2, 3 fatty acids is glycerol. It can be turned into starch and cellulose, right? Well, we talk a lot about glucose relating to starch, but we also know that cellulose is important in plants because it's the cell wall. It can also be turned into amino acids because it has a phosphate on it, and also just sugars that are monomers or dimers of polysaccharides. So those are super important, which is why G3P and the Calvin cycle are super important for plants to be able to store their food, right? Store that energy they got from the sun. If you don't create these molecules, then you don't store the energy that was originally intaken from the sunlight energy absorbed. So this is the overall process again, right? So the diagram is mainly showing you the parts of the first part of photosynthesis with photosystem 2, the electron transport chain, and then the photosystem 1, and then ATP being created because the ATP is obviously needed inside of the Calvin cycle, and ADPH accepting electrons here and then giving them away here to return back here to accept more, and the electrons being removed from water originally. So water is used here and oxygen is produced. In here, carbon dioxide is used, and then your basically your sugar base is created out of the Calvin cycle. So I want you to write these down for now, but you won't be able to answer them maybe until you get into the details of it when you watch the YouTube video for the Bozeman Science that I've posted also. If you can already answer it, that would be really, really good too, but I know that sometimes you need a couple reviews of the material to be able to, just get these points down. So you don't have to worry about filling this out now, but by the time you turn in your notes, I would like you to have this filled out after you've watched the video. And by video, I mean the Bozeman Science one, because you'll be watching this one and another video. So you get two versions of explanations. So here, scientists didn't originally know when water was being used and when carbon dioxide was being used. So they were going to do these experiments to figure out what part of the photosynthesis process are these reactants actually being used. So they tracked it by using water with a heavy isotope of oxygen, which instead of oxygen 16 is actually oxygen 18. So this oxygen is heavier, and it was given to plants. And the results were that when they had this different kind of oxygen in the water, the oxygen gas produced by the plants actually had this oxygen, and none of the oxygen in the sugar molecule that was being produced in the glucose molecule was actually this heavy isotope. So what does that mean then? What part of photosynthesis is using the water? Right, it's the part that produces the oxygen, and that would be in photosystem 2 and 1, right? Carbon dioxide with a heavy isotope of oxygen was then used to see where that was being used up, because scientists were like, where the heck is this one being used up, right? Carbon dioxide is another reactant, but we know that when you track the oxygen in the water, only oxygen gas that's produced has this heavy isotope. So then we're like, okay, so where's the oxygen going that used to be with the carbon dioxide? And when they tracked it, they found out that both the water produced during photosynthesis and the glucose molecule produced during photosynthesis had this heavy isotope when it was originally only found in the carbon dioxide. So what does this mean? Right, it means that the oxygen going into the glucose and the water molecule as a product only come from the carbon dioxide. So that means the oxygen being sourced in the products and the reactants actually have specific sources where they're coming from, okay? So what does this show? It shows over here with significance that when you have certain reactants, the products actually exclusively pull from specific reactants. So the hydrogen is obviously coming from the water in both sources, right? Because there's no hydrogen available in the carbon dioxide. But the oxygen in the water only goes to the oxygen gas being produced. And then the carbon obviously comes from the carbon dioxide, right? When you have 6 carbons, but the oxygen in carbon dioxide is going to exclusively be in the glucose and the water molecules. So that means the oxygen is not related to the carbon dioxide as a product. The product of oxygen is not related to the carbon dioxide reactant, even though there is oxygen in carbon dioxide. And the oxygen inside of the glucose and inside of the water is not related to the water at all as a reactant because they're only coming from the carbon dioxide. So this was kind of interesting for scientists to figure out, just because they were like, well, why don't they just pull from whichever one is available? And the reason is because that the water and the carbon dioxide are actually being used at separate times during photosynthesis. And that was the major discovery that they had. So let's talk a little bit about impacts of why photosynthesis is important and how it affects the environment. So we know that there's a lot of tropical rainforest destruction and climate change going on as a result of this destruction. So plants and photosynthesis are known for removing carbon dioxide, and that will slow global warming. Can you explain to me how? Write it down, and then we'll move on. So development of like human civilization and building cities and more urban areas for people to live in has reduced forest surfaces on Earth from 14% down to 16%. And what is the consequence of that? Okay. I'm going to also ask that you interpret this graph in 2 to 3 sentences. So make sure you're reading what the axes are saying, and you're looking at what happens as time has been getting closer and closer to now and the prediction for the future. Okay, and this is mean global temperature change, so it's talking about global warming and what is happening as a trend overall to the global temperature on average, and what is going to happen in the future based on scientists' predictions, based on how we are affecting the Earth. So you tell me what these are all relating to in interpreting this graph. Okie dokie. So we're going to talk about the Calvin cycle products again, just to reiterate. So when you make G3P as a result of the Calvin cycle, you're making this 3-carbon molecule, and it's connected to a phosphate because remember, like an ATP, that phosphate bond contains energy, which is easily broken and reestablished into another bond to create other molecules. So you can use G3P as glucose phosphate. You can use it to create fatty acids. You can use it to create amino acids because it does have that carbon that can be created into a ring if you combine it with another one. It also has a phosphate group that is needed in proteins. You can combine multiple of them together to make fructose phosphate, which is a disaccharide connect to a phosphate, which can then be connected to other things because the phosphate is still there, right? Because that can ultimately be turned into sucrose, which is a sugar and leaves and fruits and seeds. And most of the sugar that we consume, whether you add it as an ingredient or you can taste it in fruits and natural products, is sucrose. You can also turn it into starch, which is stored in roots, for example, in sweet potatoes and in potatoes, and also in seeds, which is used as a food for the plant seed that can help it grow, starting germination when it's replanted into the soil. And it can be turned into cellulose, which is the cell wall for all plant cells. And it's important for structure, especially in trunks and roots and branches of a plant. So let's talk shortly about the other types of photosynthesis. I'm pretty sure this is the last slide. And it looks like a lot of information, and it kind of is, but I'm just describing to you how it's different from regular photosynthesis. So CAM photosynthesis is the crusulacine acid metabolism version of photosynthesis. And this is because plants live in different environments. And in order to survive in environments that are really hot or really dry or really, really sunny or don't receive a lot of energy during the day, they have to have different methods in order to be able to still continue to make this G3P in order to survive and build cells. So CAM photosynthesis happens during the night. So CAM plants will fix carbon dioxide into an acid, okay? They'll form these C4 molecules, which are stored in large vacuoles. So they have these special packaging areas that they store this carbon 4 molecule that they've pulled from a bunch of carbon dioxide in the air. And then during the daylight, NADPH and ATP are available because photosystem 2 and photosystem 1 are currently being acted on by sunlight. So when those are available, when those are available, stomata are being closed for water conservation, right? So the water has to come from what it's already pulled before, because during the daylight, it's too hot. And if they just open their stomata, then water is going to be evaporating out. So water is being pulled in by the roots, but there's no gas exchange. So you're not exchanging carbon dioxide during the daylight. It's just too hot. They're going to dry out, right? So you're just going to do part of photosynthesis that only uses water and doesn't use carbon dioxide. And that's going to be the part that is in photosystem 2 and 1. C4 molecules are then released to carbon dioxide to the Calvin cycle once these electrons are energized during the daylight, right? So they're stored previously in the night, and then they're going to be used up during the daylight too, because that's when the daylight is energizing molecules and creating NADPH and creating ATP. And so you can do photosystem 2 and 1's process and the Calvin cycle as long as you aren't running out of those C4 molecules turning into CO2. So basically, you're pre-storing your carbon dioxide, and the next day when there is light, you're not going to be taking in any more carbon dioxide because your stomata are closed and you don't want to be evaporating out all of the water. And so you'll do photosystem 2, and then one process in photosynthesis. You'll energize those electrons, and then they will go over to do the Calvin cycle with whatever carbon molecules you have stored from the night before. Okay. Then during the following night, you'll start to store more carbon materials in order to get those carbon-4 molecules, and then those will be turned into carbon dioxide the next day during the time when photosystem 2 and 1 are active as well. Okay, so those are hot and dry climates. The reason why the stomata is closed is because it's hot and dry, and the water, if the stomata on the leaves were open, would all just evaporate straight out of the leaf, and then you wouldn't be able to get any photosynthesis done because you need the water in the first half of photosynthesis. So you're kind of, pre-preparing for that second half so that when you're doing the first half, you can also do the second half at the same time. So in hot, dry climates, there are, stomata must be closed to avoid wilting, right? Wilting, and then also ultimately the plant dying. Carbon dioxide decreases and oxygen increases as you're doing the first part of photosynthesis. Oxygen will start to combine with RUBP, and that creates a waste product, and it causes this process called photorespiration to happen, which is not producing what you want for G3P. It's going to just produce this RUBP waste product, and when that happens, then you've basically wasted all of the time that you have put into effort as a plant into creating the energized electrons, because those electrons are getting attached to this RUBP oxygen complex instead of the RUBP carbon dioxide complex. This is just because you're building up so much oxygen from doing the first half, and that's really, really bad for the plants because then they're not ending up making that carbon product that they need to make into sucrose, make into cell wall, make into amino acids, okay? So that's the problem with photorespiration and the problem with really hot climates. The C4 plants solve the problem of photorespiration by fixing the carbon dioxide to PEP, which is a molecule. You don't have to know the super details about the name. The result is an oxaloacetate, a C4 molecule, and the C4 plants avoid photorespiration. Okay, this net productivity is about 2 to 3 times greater than C3 plants, which is our normal plants, right? Because they're normally making that C3, that G3P. But in cool, moist environments, if you put a C4 plant there with C3 plants, then you actually lose out, okay? So these plants are adapted to their specific environments where it's hot and where it's dry. But when you have them in a normal climate, then the C3 plants, which are the normal plants, are actually going to outcompete them, which is why you don't find succulents in regular, cooler, drier climates. You find them only in hot and dry climates, okay? So about 95% of plants are C3 plants. So the regular plants that we see on a daily basis are all C3 plants. But when you go and you look at cacti, or you look at succulents, or you look at plants in tropical rainforests, they're adapted differently because their environments are hotter and they're either hot and moist or hot and dry. Okay, so that should be ending the notes for us now. Again, there are 3 types of photosynthesis, right? CAM photosynthesis, C4 photosynthesis, and then there's regular C3 photosynthesis for C3 plants, right? C3 is what we usually talk about when we talk about photosynthesis, and we talk about making that C3 product, which is G3P. CAM photosynthesis happens when it's hot, but there's still water available to the roots. And then where it's hot and dry, for example, in a desert, then you do C4 plants. They're a process where they're storing and they're making sure to not do photorespiration because they don't want their oxygen combining with the RUBP instead of their carbon dioxide, because then they're wasting their time and it locks the RUBP and then it can't be reused for more photosynthesis. So after this, you're going to watch your Bozeman science video and fill out that video guide and then turn it in. That's an easy free 10 points as long as you're doing it. And then if you need to, you can go back to that products reactants slide, and you're going to be able to fill this out and then turn these notes in for extra credit as well.